Welcome to episode 14 of the Pulse of Physiology podcast. I'm your host, Dr. Jim Davis, and today we're exploring the fascinating physiology of chemical synapses. These are junctions that enable neurons to communicate with other neurons or target cells. In our next episode, we'll delve into reflexes, but for now, let's focus on the intricate process of synaptic transmission. So a synapse is the junction where a neuron communicates with another cell, such as another neuron, a muscle cell, or a gland. There are two primary types of synapses. An electrical synapse, which involves direct connections between cells via something called gap junctions. These gap junctions allow ions to flow directly and depolarize adjacent cells. This form of communication is rapid and used in certain specialized functions like coordinating heart rate. And this is something that we'll talk about in more detail when we talk about cardiac pacemaker cells and the contraction of the heart. Then we have chemical synapses. In this more common type, neurotransmitters are released by the presynaptic cell. They cross a small gap called the synaptic cleft and then bind to receptors on the postsynaptic cell. This process is slightly slower but allows for more precise and flexible control. Now, there are several key structures involved in a chemical synapse. These include the axon terminal, sometimes referred to as the synaptic knob, and this is the end of the presynaptic neuron where neurotransmitters are stored in vesicles or containers, or which are a type of container. Next, we have voltage-gated calcium channels. These channels open in response to an action potential and ultimately allow calcium ions to enter the presynaptic terminal. Synaptic vesicles are these small sacs or containers that contain neurotransmitters. The synaptic cleft is the narrow gap between the presynaptic and postsynaptic cells. And then finally, we have neurotransmitter receptors, which are proteins on the postsynaptic membrane that bind neurotransmitters and ultimately trigger a response in the target cell. So synaptic transmission occurs in a series of carefully orchestrated steps. To start off, we have to have the arrival of an action potential. When an action potential reaches the axon terminal, it depolarizes the cell membrane. This depolarization allows for calcium influx because voltage-gated calcium channels open. So calcium ions flow into the presynaptic cell. Now, when calcium flows into the cell, it binds to synaptic vesicles. This binding triggers the vesicles to move to the membrane and release their neurotransmitters into the synaptic cleft. This is the process of exocytosis. Once the neurons are released, they diffuse across the synaptic cleft and bind to receptors on the postsynaptic cell. Ultimately, and depending on the receptor type, the binding of these neurotransmitters to the postsynaptic cell can either excite, also known as depolarize, or inhibit, also known as hyperpolarize, the postsynaptic cell. This leads us to agonists and antagonists. Now, neurotransmitters can have different effects depending on their interactions with receptors. We can have agonists. Now, agonists are substances that activate a receptor that cause the ligand-gated ion channel to open and initiate a response. Think of an agonist as your friend that's willing to help you out. Then you've got antagonists. Antagonists are substances that also bind to receptors but prevent activation, keeping the ligand-gated ion channel closed. So this would be like your enemy or your little annoying brother or something like that that prevents you from doing the thing you want to do. Generally speaking, neurotransmitters are not agonists or antagonists for each other. They're just nothing when it comes to this kind of relationship, and that's because of their different chemical structure. So when we are referring to agonists and antagonists, we're talking about specific substances that have similar shapes and can basically fit into the same keyhole. So ultimately, once we have a signal sent from the presynaptic neuron to the postsynaptic cell, we need to end this process. In other words, we don't want the presynaptic neuron to constantly stimulate the postsynaptic cell, so we need to terminate this activity. And so this occurs through several ways. First, we have diffusion. Neurotransmitters will drift away from the synaptic cleft into the surrounding extracellular fluid. And if they're not bound to a receptor, they're not going to cause any changes. Next, we have enzymatic breakdown. So this is where enzymes degrade or break down neurotransmitters, ultimately rendering these neurotransmitters inactive. For example, acetylcholine is broken down by the enzyme acetylcholinesterase. Finally, we have reuptake. Neurotransmitters are reabsorbed by the presynaptic neuron from where they came from, or other cells such as glial cells, which will use them or which will recycle these neurotransmitters. So you might be asking yourself, why are chemical synapses important? Well, ultimately, they provide the nervous system with incredible flexibility and precision. They allow neurons to integrate multiple signals, modify responses, and coordinate complex functions like learning, memory, and muscle contraction. So let's summarize with our key takeaways. A synapse is the junction between a neuron and another cell, which facilitates communication. Chemical synapses involve neurotransmitter release, diffusion, and binding to receptors on the postsynaptic cell. Synaptic transmission occurs in distinct steps, including calcium influx, exocytosis, and receptor activation. Neurotransmitter activity is terminated through diffusion, enzymatic breakdown, or reuptake. And so with that, we are all finished with episode 14. I hope this episode has provided a clear understanding of how chemical synapses work. Join me next time where we'll discuss reflexes and their role in the nervous system. Until then, keep questioning, keep exploring, and stay curious.